Hello team! So today I'm making a short video about the neuromuscular junction and the sliding filament theory. This is going pretty in depth about how this happens within a cell and what receptors and ions and all are all involved. So if you can see on the side here, I'm just getting my markers ready. I am also posting an outline to go along with this, and this will be a good self-check for you to know if you know this process really well, because this is the meat and potatoes of the cell and the muscular system. And it all begins with the neuromuscular junction and the motor unit. And the motor unit is really just your lower motor neuron and the muscle fiber it innervates. So for example, this is what we have here. So we have a lower motor neuron and it's innervating an example muscle fiber. Whee! And of course it's a synapse, so it has that little gap of space. It's not actually touching it. So what happens when this lower motor neuron is told that, hey, this muscle fiber needs to contract, what happens is this neuron is going to exocytose acetylcholine. And on the muscle fiber, what we have are little nicotinic acetylcholine receptors. And these are voltage gated sodium channels. So when acetylcholine is exocytosed, acetylcholine will bind to these. And to get why this is important, we're going to go take another step and think about the T tubules. And we can model these as just sort of a little dent in a road. You can imagine you're walking along the sarcolemma and you're going to fall into a little T tubule. And this makes a little bit more sense with the diagram in your book. If you can see here, you have your sarcoplasmic reticulum and your sarcolemma. And these T tubules are just invaginations in those. They're literally transverse tubes in your muscle fiber, which is pretty dang cool. So let's go ahead and erase that guy because some really crazy stuff is about to go down. So all along your T tubules, you have those voltage-gated sodium channels. Because they're voltage-gated and you had sodium come in because of that acetylcholine binding, you have sodium in, coming into the cell. What happens is sodium is going to keep going along these, whee, and causing an action potential and membrane excitability. And this is really important because on that membrane, getting excited is our good friend dihydropyridine. And we abbreviate dihydropyridine to DHP. And when DHP gets excited, it moves backwards a little bit. So we can imagine it going kind of back here instead of up there. So why is that important? Well, because within our membrane here, we have the sarcoplasmic reticulum, which is again a modified sort of endoplasmic reticulum. And in these muscle fibers, it's a big bag of calcium ions, Che+, my bad. And on the sarcoplasmic reticulum, you have what we call ryanodine receptors. And we're going to label these in green here, and they're abbreviated RYR. So sodium has passed, is excited, and it's actually caused this dihydropyridine to unplug from ryanodine. If there wasn't any sodium, normally dihydropyridine is plugging up this ryanodine receptor's channels and it can't allow calcium to go through. But when we do have sodium, the membrane is excited, dihydropyridine will move back, and this is all because acetylcholine has binded to this nicotinic acetylcholine receptor. And this is going to allow calcium to flow out of the sarcoplasmic reticulum and into the cell. And what's really important is within our cell, remember, we have myosin and actin, our star-crossed lovers. So we have our M line and our myosin filaments here. These are our thick filaments. And then on the other side, each side of them, we have our Z line with actin, which are our thin filaments. Whee! And they go in right between those myosin heads. Draw a quick Z line there again. And so this is our functional unit in the muscular system for contraction called the sarcomere. And these sarcomeres individually will shorten during contraction. And the summation of all these contractions of sarcomeres is what causes a full muscle fiber, muscle contraction, causes the shortening of your muscles and all that good stuff we've already talked about in the gross physiology of the system. So let's take a closer look at our thin filaments and thick filaments. So we have our thin filaments, which are actin, and they're basically just little blobs of stuff. And if we want to go really in depth, we can talk about how we have G-actin, which are these little globes, and in between them we have like a little line of what we call F-actin, which is filamentous actin. It sort of keeps those glomerular G-actins in place. And on our actins, we have active sites. On G-actin to be specific. So we have all these little active sites, but most of the time these active sites are covered by a good bodyguard friend of actin called tropomyosin, which we're going to represent by this blue ribbon. I can't draw 3D as well as the book represents it, but you can see the same idea on the same page actually of your book. You have your tropomyosin here, you have your G-actin. They don't actually have filamentous actin shown, but that's okay. You get the idea. Tropomyosin is covering the active sites on actin. And this really sucks because we have myosin here, which is our thick filament, with its little tail, its hinge, and then its head, which kind of looks like a little golf club. And it really, really wants to bind to those active sites on actin, but it can't because tropomyosin is covering. Whatever will it do? Oh, this is exactly where calcium comes back in. So calcium is flowing into the cell, and we have our sarcomeres, and what happens is on tropomyosin, we have a special protein called troponin. And what happens is this troponin loves to bind to calcium. And so what happens is this calcium is going to flow out, and this calcium is going to make a beeline. It's going to make a beeline straight to that troponin. And remember, when proteins bind to stuff, proteins will change shape. And so what happens is troponin is happy as calcium binded to it, and it's going to move our tropomyosin, and we'll just sort of represent this as it maybe on top here. So we have our tropomyosin moved because calcium has bound to troponin, and it exposes actin in all its glory, and especially those active sites. So myosin is super excited. It gets to bind finally with actin, but it's not quite there yet. In order for myosin to bind, what needs to happen is it will have ATP bound to it as well. But once myosin can convert that ATP into ADP plus inorganic phosphate, which we abbreviate to pi, then it can reach up, can move its little head, and it will bind to that active site on actin. So what happens, so it has the ADP and pi, and what will happen is it'll actually just sort of move that actin. It'll start to contract. That myosin head is moving it, and it's going to sort of pull its head this way. And what happens when it pulls its head that way, the ADP and inorganic phosphate unbind and instead leaves rooms for another ATP to bind. So we can imagine that it's moved this actin this way. But when that ATP binds after myosin has pulled that actin closer to the M line, that's the idea, it's moving the Z lines towards the M lines. Once myosin has done that, it's going to have that ATP bind. When that ATP binds, remember, when stuff binds to proteins, proteins like to change shape. And what happens is myosin will have to unbind with its lover actin, and it's going to go back to where it was. It's going to go back, and it's going to be sad. It's going to be, oh no, I need to convert ATP into ADP and pi again so I can be with my lover actin once more. And this will continue. The ATP and pi, or the ATP will convert to ADP and pi. It'll bring it close to the M line, and it'll just repeat until the muscle is ready to stop contracting. And of course, when the muscle is ready to stop contracting, it goes all the way back to this neuron innervating this muscle fiber. So when the muscle is ready to stop contracting, acetylcholine will stop being produced. That means sodium will stop flowing into the cell with these voltage-gated channels, and that will unexcite the dihydrogen pyridine, which means it's going to want to close up on that ryanodine again, which means ryanodine doesn't want, all of its calcium being depleted. But what it'll do is it means all that calcium is going to start flowing back into the sarcoplasmic reticulum. All that calcium is going to unbind from troponin, and it's going to cause tropomyosin to move back and cover all those active sites on actin, and troponin goes back to being sad because calcium is no longer bound to it. So everything essentially goes back to normal instead of being contracted. So our M lines don't really change, but what will happen is actin will get closer and shorten that sarcomere. But it'll go back once all this happens to its regular length, its resting length, essentially. And that's the basics of the neuromuscular junction and sliding filament theory. So I hope you guys enjoyed this. I hope you guys use the outline, and I hope you guys can use this video as a tool. So maybe if there's a certain part that was confusing, you can go back, rewatch it. You can go to the outline and see what it says, or you can also ask me questions during our next meeting if you need some extra clarification. I'm always willing to help. I hope you guys enjoyed and have a great week.